Alright, and I left off on this slide in the last video, and I wanted to finish up that, of course, we're looking at this in lab as well. The pharynx is actually 3 basic regions, and you have your nasopharynx, which is from the back of the nasal cavity to about the uvula, which marks the soft palate. And then the oro is from that uvula to the epiglottis. And then the laryngopharynx is from that epiglottis down to the esophagus, right about here. Okay, so we'll be looking at that in lab as well. The esophagus then is going to, there's an upper esophageal sphincter and a lower one. We also call this one the gastroesophageal sphincter, and sometimes it's called the cardiac sphincter. Right? So these are the guardians of the tube itself. So as you look at the slide in your lab, you'll notice that the esophagus, or if you recall it from looking at it in AP1 when we did stratified squamous epithelium, it's all collapsed, if you will, when there's no food in it, which shows that it can expand. So there's also stratified squamous, and then there's lots and lots of elastic fibers in here. So the esophagus runs from the laryngopharynx down to the stomach, so passes through the diaphragm. So it moves from the mediastinum area so right about here where the heart is found, down through the diaphragm, a hiatus in the diaphragm. And that's where you get hiatal hernias, when a little bit of the esophagus can be pushed backwards through that hole. So when you swallow, right, and this whole structure is for just moving it. When you swallow, that upper sphincter relaxes, and the lower one will as well, to open up. So these are, again, sphincters are thickenings in the muscularis, the circular muscularis. And you'll have that wave of the circular relaxing in front and contracting behind it. And then the longitudinal muscle that lines this tract will push. So we are going to see from esophagus all the way down through the rest of the tubing, these layers that we talked about on the previous video. All right, so here's a nice histology of this to get you an idea of again, it's all folded up. This is very low magnification. So this is all muscle out here. This would be the submucosa with all kinds of glands that will just secrete mucus to basically lubricate food as it moves through this, okay? So stratified squamous, just like the oral cavity was, has mucus-secreting glands in this connective tissue we call the lamina propria. And then the muscularis will transition from skeletal to smooth muscle, which again, there is some skeletal, but it is all really out of your control. It's an involuntary response to move food through the esophagus. All right, deglutition is what we call swallowing. And so we know this is going to happen at the posterior of the oral cavity and all the way down through the esophagus. So this kind of just walks you through. There are 3 basic stages of the swallowing process that which you choose, right, voluntary, to begin this. So in the oral cavity, pushing it to the tongue, all because it's sensory. And once you get it to a position on the posterior tongue, the pharyngeal stage will begin, all right? So as it reaches the oropharynx by that soft palate, it's controlled by the swallowing center in the medulla. And what happens is you will elevate really the soft palate to block the nasopharynx. And the larynx will rise up, will move upwards, and the epiglottis will move downwards to cap or prevent food from going into the trachea. And there we will start this constricting and relaxing of muscles to push the bolus of food to the esophagus. The esophageal is also involuntary, and this is when food enters the esophagus and muscles again contract and relax to undergo peristalsis and propel it to the stomach. And also glands are secreting, which is, again, out of your control. So once we're in the stomach, this is basically a esophagus to duodenum, resting area or holding chamber. I say you can eat faster than, sorry, I misspelled that, then your small intestine can digest and absorb, so the stomach holds and then permits small amounts to move into the intestines. So this is why we say the stomach is really a holding chamber. And so in the stomach, mechanical digestion will continue because we have not yet gotten food to the form it needs to be for the best or most effective chemical digestion. And the chemical digestion will continue. So we started some carbohydrates with amylase in the mouth. And while we secreted some lipases for lipids, they really haven't been active yet. And here in the stomach, we're mainly going to target our proteins, right? So we'll stop the function of amylase, so there'll be very little chemical digestion of carbohydrates that will continue in the stomach. We will begin the chemical digestion of proteins, and we'll do a little bit of the lipids. The stomach also produces a chemical called intrinsic factor, which allows us to effectively absorb B12. Right? And remember, we need B12 to make red blood cells, as well as use it for some of our other metabolic processes. In the stomach, we're going to convert the bolus to chyme. There's not going to be much absorption, and then we're going to transport it to the small intestine. So it's gross anatomy. It has a number of different regions: the cardiac region, the fundus, the body, and the pylorus. It's got these curvatures that you look at in the lab, and then of course it has that but this gastroesophageal sphincter, or cardiac sphincter, and the pyloric sphincter, which is often very well developed in the stomach. The stomach has additional layers of muscle. There's an oblique layer, so it's a highly muscular organ, and this is what's going to really help it with that mechanical liquefying of food. All right. When it's empty, you see these massive, large folds in the mucosa called rugae, or gastric folds, and this signifies that distensibility I was talking about up here. So it is a highly distendable or stretchable holding chamber. Right? Histologically, some of the differences we see, well, now we're going to start the simple columnar that you see here and then magnified here. And we have pits in the stomach. So in the rugae are these big, gentle folds in here. But if we stretched out the stomach, right, and looked at the mucosa specifically, it is basically comprised of a whole lot of these pits, all right? Now, towards the surface, where the food would be right in here, would be these mucus neck cells, which would, again, produce a lot of mucus to help protect the cells here and the cells of the stomach from the stomach's own secretions. In the pits, we would see what we call the gastric glands. All right, so moving down into the pit are these glands. And in these glands, we see, right, as I show you here, we magnify parietal cells, chief cells, and enteroendocrine cells. So 3 different cell types that are very important for function. And then we have our submucosa, we have the layers of the muscularis, and the serosa, as we said, would be there. All right. So these goblet at the surface epithelium and part of lining the pits are a lot of mucosal cells that really produce this alkaline, so protective, buffering mucus. And these cells are replaced at least weekly. The pits to the gastric glands, and the glands produce what we call gastric juice, while the pits are, again, surface mucus cells with lots of mucus for protection. And the glands are parietal cells, enteroendocrine chief cells, and again, those mucus neck cells. So here, when we look at the specific cell types, which we want you to know that the parietal cells make hydrochloric acid, okay? And hydrochloric acid is what we're going to use to activate pepsinogen, right? So there's the OGEN, which means this compound is not active yet. And in the presence of hydrochloric acid, pepsinogen made by chief cells becomes pepsin, right? So activates pepsin, denatures proteins, so acid, right, unravel these proteins. That's why when you did your OT1 experiment on the enzyme catalase, and you made the whole test tube highly acidic, the enzyme didn't work anymore. So it didn't work because you had denatured it. So now pepsin itself is specialized to work in an acid environment, but remember that a lot of what you're eating are just basic proteins. Proteins in your food, right? Lipids in your food, carbohydrates in your food, nucleic acids in your food. These are the 4 major groups. And here in the stomach, we're going to really specialize at trying to access the proteins. So proteins get denatured, so when they become denatured, that makes them unwind or unravel, unfold, and that allows enzymes to be more effective. Because bacteria are made of proteins, when the secretions of the parietal cells are released into the stomach, it will kill the bacteria because it destroys the proteins. And then intrinsic factor is also made by the parietal cells, and this is for B12 absorption. Chief cells in there make pepsinogen, the main enzyme, and again, this digests proteins. And then enteroendocrine cells, well, they're the source of gastrin we mentioned earlier, right? And then there's some other hormones actually made by these cells, but gastrin is the main one and the one we want to know. But if you look at table 23.6 on page 1110, you will see what these other hormones are responsible for. All right, so regulation of these gastric secretions, right? So we just went through what they were and who made them, so let's figure out how they get controlled so that they do the most effective job. And here again, we're thinking neural hormonal, right? So I say this is an interaction of the brain, the stomach, and the small intestines, and that's why we divide gastric secretions or the gastric process of digestion into the cephalic phase, the gastric phase, and the intestinal phase. And these are named for what part of the body is in charge, right? So I say the neural aspects are the higher brain in control, local reflexes in the enteric nervous system, and the autonomic, parasympathetic stimulating, sympathetic inhibiting. Hormonal regulation is done through gastrin, which stimulates all aspects in the stomach, or gastric inhibitory peptide, cholecystokinin, and secretin, which inhibit what's happening in the stomach. Okay, so here again, we want to first talk about driving, promoting, stimulating the whole gastric process. So that would be the cephalic phase and gastric phase. So I say here, sight, thought of food, smell, right? And it's your higher brain talking to your emotional brain and then sending information down to the stomach to say, hey, start making the substances, a little bit of hydrochloric acid, gastrin, right? Start getting these glands ready. Okay, and then here's the the inhibitory aspect. So when you're depressed, loss of appetite, lack of stimulation would lead to that. But here we're trying to drive the digestive process. So let's look at this. All right, gastric phase. So once food enters the stomach, so this is stimulated by these things. Once the gastric phase is stimulated by stomach distension, right? Those stretch receptors, and then those chemoreceptors here, which sense the chemicals, the chemistry of our food, and an increasing pH. So both of these sensory aspects will lead to, right, more gastrin being made and dumped into the blood and stimulating all the secretory activity. So the parietal cells, the chief cells. And then these local reflexes too that are sensed by this distension, which will also stimulate more of the secretory. All right. In the intestinal phase, we're actually going to have the small intestine in control, and it's going to work to try to make sure that the food entering the small intestine is appropriately digested and the pH is appropriate for the work of the small intestines. So this stage is usually considered more inhibitory, so we have to focus on this box. So we say distension of the duodenum, presence of fatty, acidic, hypertonic, so high concentration chyme. Right? Done through local reflexes, the pyloric sphincter is tightened, and we call this the enterogastric reflex. And the again, the small intestine is telling the stomach to slow down. Right? Distension, presence of fatty acids and partially digested food will cause the release of secretin and cholecystokinin and gastric inhibitory. So these guys, which I said inhibit the process of gastric digestion, will become stimulated during the intestinal phase, right? And we're going to slow down what the stomach is doing here and try to make it appropriate for the small intestines, right? And we will stop there. I know this is a long explanation, and continue on in the next slide.